The cell is the simplest collection of matter that can function all by itself. Microscopes allow us to study cells, and even smaller structures that are found within cells. Microscopes are used to magnify an object, or to make an object appear larger than it actually is. And the ability of a microscope to provide a clear magnified image relies on two attributes: contrast and resolution. Contrast is the relative difference between light and dark. It's the property that allows you to distinguish objects from one another and from the background based on color and brightness. In this image, the left-hand side is at a low contrast and the right-hand side is a high contrast image. You can see that it's much easier to distinguish between objects in the background in the high contrast image. Resolution is the measure of clarity of an image. It's the minimum distance two points can be separated and still be distinguished as 2 points. For example, in the image at the bottom, on the left, the figure appears to be one elongated spot, while at the higher resolution on the right, two spots are easily distinguishable from one another. The resolution of a microscope is limited by the shortest wavelength of light used to illuminate the specimen. The shorter the wavelength, the higher the resolution. And the higher the resolution, the smaller objects you can clearly see. A light microscope uses visible light to pass through a specimen mounted on a slide, and the image is magnified using a series of optical lenses. Because a light microscope uses visible light, it can only magnify a specimen to about 1,000 times its actual size. If you go beyond that, the resolution and the contrast of the image is lost, and you lose a lot of the detail in the image. Because of these limitations, a light microscope can provide a clear image of objects only as small as plant and animal cells and most bacterial cells. A big advancement with microscopes came with the invention of the electron microscope, which instead of light uses a beam of electrons. That beam of electrons has a much shorter wavelength than visible light, so it provides a much higher resolution at a higher magnification. This allows electron microscopes to see objects as small as ribosomes, proteins, and lipids. There are two types of electron microscopes: scanning and transmission. Scanning electron microscopes are very useful for the study of the surface of a cell. The electron beam scans the surface, which is usually coated with a thin film of gold. That beam excites electrons on the surface of the cell, and those secondary excited electrons are detected by a device that translates the pattern of electrons into an image. Transmission electron microscopes are used to study internal components of cells. The electron beam is passed through a thin section of a specimen that's been stained with atoms of a heavy metal that only bind to certain structures within the cell. And again, the pattern of electrons that's transmitted through the cell is detected and translated by a device into an image.